What are mutations? Mutations lead to genetic variation or differences among species. Mutations are changes or differences in the DNA base sequence. For example, if there was the base adenine A in the sequence and then it was substituted by cytosine C. Mutations are completely random, meaning that they are not intentionally planned to occur. Mutations can be beneficial, such as a trait that helps an organism to survive. They can be harmful by causing a disease, or they could be neutral or unnoticed in the physical appearance of the organism. Mutations can take place in body cells or in the sex cells. Mutations will only be passed on to the organism's offspring if they take place in a sex cell. There are two ways in which mutations come about. The first cause is an external cause or environmental factor. For example, exposure to chemicals or radiation can damage the DNA molecule. Cigarettes contain chemicals that damage the cells. These chemicals are called carcinogens and can lead to cancer. The second major cause of mutations occurs during DNA replication, when the DNA is making a copy of itself before it divides into two new cells. These mutations are natural and cannot be prevented. They lead to small changes in the DNA sequence. The cell cycle is the process of cells growing and dividing. The first stage, interphase, is the stage before cell division where the cell grows to its mature size. Then the cell makes a copy of its DNA during DNA replication. The cell prepares to divide into two cells during interphase. By the end of this phase, the cell is ready to divide. Interphase is also the longest part of the cell cycle. After learning about interphase, you might be wondering what happens during DNA replication. The double helix unwinds like a zipper and separates into two strands. New bases pair with the matching bases on each strand to form pairs identical to the original strand. And as a result, two identical DNA molecules are formed. The order of the bases in the new strand will match the order of the original DNA molecule. The important thing or key message to take away from DNA replication is that two identical DNA molecules are formed. Each of the new molecules has one of the original strands of the double helix. During the second stage of the cell cycle, mitosis, the cell's nucleus divides into two nuclei. One copy of the DNA is distributed to each of the two daughter cells. Mitosis is divided into four parts, called phases. In the final phase of mitosis, the genetic material moves to opposite ends of the cell. Two new nuclei form, one at each end of the cell. The final stage of the cell cycle is cytokinesis. During cytokinesis, the cell completes the process of cell division. The cell membrane pinches in the middle of the cell. During this stage, the cytoplasm also divides and distributes half of the organelles to each of the daughter cells. The new cells will continue the cycle and enter interphase. As you can see, the cell cycle repeats itself over and over again. One thing to note is that plant cells differ from animal cells because they have a rigid cell wall. They form a cell plate structured across the middle of the cell during cytokinesis before they divide. As I had mentioned earlier, mutations are the changes or differences in the cell cycle or changes in the DNA of a cell. Mutations can cause cells to function abnormally. Normal cells regulate when they need to divide to create more cells. Damaged cells sometimes cannot regulate when to start or stop dividing. This uncontrollable dividing is called cancer. Cancer occurs when the cell cycle is disrupted. In the pictures below, you can see normal cells and where a tumor or a group of cancer cells is forming. These cancer cells begin to invade other parts of the body and take their spot. In the next few days, we'll be taking a closer look at mutations.